Welcome to today's Before You Buy Q&A session. The Before You Buy video series takes a deep dive into the essentials of laboratory equipment purchasing with the help of an industry expert. Today, we're joined by Sheela Muley, Product Manager, BioPharma and Molecular Devices, who will discuss emerging automation solutions for microbiome research and development. Hi, Sheela.
Hi, Damon.
Microbiome research involves labor-intensive tasks such as colony picking, plating, liquid handling, and streaking, all of which demand high levels of precision, sterility, and repeatability. To add to these challenges, most commercially available automated colony pickers and liquid handling systems are designed for use in ambient laboratory conditions. The too large or unsuitable for placement inside controlled atmospheric chambers. The field of microbiome research is advancing rapidly, but isolating and culturing the right strains, especially under anaerobic conditions, remains a major bottleneck to workflow efficiency. So in today's discussion, we will explore how automation is reshaping microbiome research workflows, the current landscape of automated colony picking solutions, how the QPix FLEX automated microbial colony picker can improve efficiency and support lab sustainability, and how instrument solutions like the QPix FLEX can help drive the future of microbiome investigations. So a little bit about our guest. Sheela is a seasoned biopharma product leader with over 20 years of experience in analytical platform development, high-throughput screening, and commercialization of life sciences technologies. She currently leads BioPharma product portfolio at Molecular Devices, where she has driven new product initiatives on microbial and mammalian cell cloning screening solutions. Let's start with a top-level question for you. What are the major challenges associated with modern microbiome research, Sheela?
Well, thanks, Damon, for that question. Before I get into the challenges, I want to give a little bit of background on what our gut microbiota is and dwell into how it affects everything about us. So the human gut microbiota is a vast and complex ecosystem. We're literally home to trillions of microbes that collectively call us their homes and encode over 150x more genes than our own human genome. So making us more microbes than human at a genetic level. So these microbes play a critical role in regulating metabolism, immunity, and even brain function. A growing body of research links the microbiota to a wide range of diseases. For example, diabetes, anxiety, depression, Alzheimer's, and even Parkinson's. While there have been early therapies like fecal microbiota transplant or FMTs, they showed promise, particularly around recurrent C. difficile infections. They faced challenges with safety and consistency. Then there were second-generation therapies, which focused on culturable species, offer improved safety, but are limited by the fact that only 37% of our gut microbiome is culturable, bringing you down to what you talked about initially about the challenging environment that these need to grow into. So this leaves about 60 to 70%, which is a majority of the microbial diversity in our gut microbiota, is still unexplored. So the third generation of approaches that are currently emerging, they leverage genome-guided culturing and AI to unlock this hidden diversity. While next-generation sequencing, or NGS, can tell us who these microbes are, but what they don't reveal are what they do and how they interact with the host, which is us in this case, and how do they transform our bodies and our mind in this unexplainable manner. So culturing these organisms, which are many of the times are strict anaerobes, remain a major challenge. Isolating, growing, and tracking them requires extreme precision and care. And so the current workflows in anaerobic chambers are largely manual. One, because there isn't an instrument that can take care of all the steps of the workflows. So being manual, it's also slow and inefficient. And because of this, that everyone is using manual processes, there's a lack of standardized protocols for culturing, formulation, and delivery. And without this foundational infrastructure, progress in microbiome therapeutics remain fragmented. So that's the reason there are so many challenges with this in this area.
Excellent. Yes, great. Thank you very much for the overview. And it leads to my next question. How is automation changing the landscape and helping to address these challenges?
Yeah, so automation is pivotal, right? Particularly in culturing and characterizing anaerobic organisms. By enabling automated workflows within anaerobic chambers, researchers can now bypass the limitation of manual culturing, reducing fatigue, human error, and significantly improving efficiency. Automation also tends to standardize protocols, which is a challenge in this field, which enhances reproducibility and throughput across the labs. These automated systems can streamline data management and data generation and storage, and creating structured data sets that are ideal for machine learning integration. So as with the advent of machine learning and AI, we want to be utilizing that more and more to advance our therapeutics, right? So this allows our researchers to extract biologically meaningful patterns that would be difficult to detect manually. As a result, scientists can shift their focus from labor-intensive data collection to high-value insight generation, accelerating discovery and enabling more targeted functional exploration of microbiome rather than just the manual work.
Excellent. An amazing emerging area of technology. And I know that there are a number of solutions that are coming out that are currently in the market. So maybe you can describe some of the different automated colony picking solutions that are out there, and maybe even go into some pros and cons, what they're able to do, and then what are some insufficiencies that can be improved upon.
Absolutely. That's a great question. So the current landscape of automated colony picking, it is evolving rapidly, just with the evolving scientific arena, and it's driven by the growing complexity of microbiome and synthetic biology research. So most systems on the market currently are designed to automate high-throughput colony selection, replacing manual repetitive tasks with faster, more consistent picking. While these tools are effective at increasing speed and reducing labor, they often function as a standalone pickers with limited integration into broader workflows, which is important for microbiome. If you're working inside a challenging environment, you don't want to be working with a fragmented workflow. They typically lack the flexibility to support this multi-step processes, such as starting from media dispensing to plating or streaking, advanced phenotype-based selection, because you are now working with non-model organisms, whereas the current systems on market are mostly used to working with model organisms like E. coli or yeast. And the operation inside an anaerobic or challenging environment, the stability of the system itself within that, right? So additionally, many of these systems may fall short in areas like sterility control, traceability, and ease of use or maintenance. factors that are increasingly critical when working with non-model organisms, as I said before, and also which are typical of microbiome research. In contrast, the field is shifting towards more precision-driven integrated systems that enable more strategic data-informed workflows. With the integration of AI ML technologies, researchers are now moving away from blindly screening thousands of colonies to focusing on those with specific phenotypes or predicted functions. This shift supports more efficient, biologically meaningful discovery, no longer searching for a needle in a haystack, but instead collecting the diversity needed to drive deeper insights and accelerate therapeutic development. So, going into some of the pros and cons, like I said, any of these systems that are currently robust in terms of colony picking solutions, they are mid to high throughput, but as I said, they only function as one single standalone system. So instead of integrating the entire workflow, for example, some of these may be big and bulky, as you mentioned earlier, Damon, in the talk. And some of these are hard to operate or place inside an anaerobic chambers. They may or may not have air compressors, which are not compatible with strict anaerobes. They may or may not contain like halogen heating for sterilization, which is again not compatible with anaerobic research where hydrogen is involved. So getting all of these factors together, they are sort of the limitations of current existing technologies.
Excellent. It sounds like these technologies can do a task or two. They have a limited reach for the tasks they can accomplish, but they cannot be an end-to-end solution.
Yeah.
And it also sounds like there's an issue with integration into workflows, which is so important because when you're working with precious samples or difficult to culture more microorganisms, you need to be able to integrate into your workflows, and you need repeatability and you need standardization of your protocols. So it's so important to have an end-to-end solution, right?
Yep.
So anyone looking to embrace this new level of technology and these new features and automation-friendly capabilities that are coming out, what are some things they should probably keep in mind in order to maximize their investment?
So some of the solutions, it is essential to look beyond just a colony picking. As we talked about earlier, consider how well the system integrates in your broader research pipeline. Key aspects should support multi-step workflow, right? Especially when you're placing a system within a hypoxic or an anaerobic chamber, you want to be able to perform multiple steps within that. You cannot have 10 systems sitting in that anaerobic chamber, one doing liquid handling, the other one picking, the third one doing hit picking. So all of these processes need to be combined. So thinking about more streamlined or optimized workflow solution is a key step. Also, compatibility and stability of these systems. I mean, you can take a liquid handler and put it inside an anaerobic chamber, but does that mean it's going to function reproducibly? That's something that you need to look at. Obviously, sterility is very important. And traceability, sample tracking over time, ease of use, especially in that awkward environment, right? If you're trying to reach in, you don't want to have complex calibration that would require you to go in and look at the instrument every day, calibrate in a certain way. So that's also important. And again, reproducibility is the most important process as well, because without that, you do not have a good lab practice. So that's also important. So some of these considerations, I would say.
Excellent. And as far as new technology, can you detail some of the features and advantages of new technologies like the QPix FLEX instrument? Obviously, you're an expert in this area, and you can tell us a little bit about its ability to address the major bottlenecks that we discussed, and help push the field forward.
Yeah. So QPix FLEX, this exemplifies the next generation of solutions. It is more than a colony picker. It is an intelligent automation platform that basically combines plating and streaking, colony picking, hit picking, and liquid handling in one compact device. It supports phenotype-based selection, operates within an anaerobic chamber, and ensures full traceability through integrated barcode tracking and data management. Unlike traditional systems, QPix FLEX enables true workflow streamlining by bridging the upstream culturing with downstream analysis, with the data output and the ease to export the data, as well as keeping everything integrated and making it as a cornerstone for targeted reproduced microbiome, as well as synthetic biology research. So it's not just one workflow. It actually, you can use it for multi-verticals. Some of the features of QPix FLEX that I want to discuss today is one of the first and foremost features is its multifunctionality. It's, as I said before, it's not just a colony picker. It's a liquid handler as well as a colony picker all in one. So it can do your plating and streaking as well as hit picking along with colony picking, and colony picking with advanced morphology as well as color-based imaging. It can do simultaneous sample and control picking, agar-to-agar transfer, and it also has the flexibility to seamlessly switch between sterilizable and disposable tips. It has a very user-friendly deck layout, very adaptable. It can support a variety of labware. We also have features like which gives us greater than 99% accuracy, like ultrasonic sensor, which is a multi-point ultrasonic sensor. And then there's a high precision mechanical stepper. It's, as I mentioned before, it's compact size that can be contained within an anaerobic chamber, or if you want to simply put it on your lab bench, you can do that as well. We have also verified its durability for in the presence of 5% hydrogen for close to 1400 hours, which for a typical use, it's 1.8 years of a typical use. And the sterility, we have multiple options for sterility as well. There's UV sterilization. We have the ultrasonic bath washing for sterilizable tips, as well as optional HEPA. The traceability is achieved via 2D barcode reading as well. So that keeps everything like integration with all the processes as well as the data that you can then take and generate insights.
Excellent. Thank you for taking us through all the different features. And, a device like this helps streamline workflows by merging all of the individual steps. And I imagine there's the workflow efficiency concept, but there's also the sustainability concept, meaning less resource usage is used because you're merging these different workflows. So maybe you can talk a little bit about maybe the sustainability aspect, because it is something that's on the mind of anyone in the research environment.
Yeah, absolutely. And as I said, like within this, as I mentioned during the features, like we also have the ability to switch between sterilizable tips as well as disposable tips. So anyone who's looking for eco-friendly solution, they can use sterilizable tips as well. Obviously, within this, what we have seen with our some of our beta testing customers is they were able to pick really small colonies, about 0.2 millimeters, which saved them about 1 to 3 days, which is more sustainable, as well as they were able to, their throughput increased by 3x. So all of these factors combined together, when you're looking at smaller colonies, that also means that you're able to look at more diversity at the same time. You are basically, if you have contamination or if you have species that can overgrow the ones that you're looking for, you have the ability to pick those up early. So that way you have the ability to collect more diversity as well. So all of these factors actually improve productivity, and that combined with ability to have sterilizable tips improves sustainability.
Yeah, I would imagine too that having the sensitivity and accuracy up front allows you to pick the right colonies and move through a workflow rather than having to go back to the beginning. And so right there, you're preserving a large amount of resources just by having that accuracy, that ability to do it right the first time. So taking a step back, in your view, what do you think the future of microbiome research looks like?
It's advancing. For example, there's been decades of research where we know, based on NGS data and all of that, we know that our microbiome affects a lot of our susceptibility to a lot of different diseases. So the future is shifting from just cataloging this microbial diversity to actually decoding microbial function and its impact on human health. Powered by AI, machine learning tools, and multi-omics integration, the field is moving more towards precision, as well as microbiome science, where microbial signatures can guide personalized therapies, diagnostics, as well as disease prevention, rather than just saying there's correlation. Now we're going towards causation. Researchers are increasingly focused on targeted phenotypic-driven discovery rather than just high-throughput screening. Supported by intelligent automation platforms like QPix FLEX, these complex workflows can be streamlined as we gain deeper insights into host-microbe interactions. The microbiome field is poised to become the cornerstone of next generation of therapeutics, spanning gut health, metabolic disorders, immunology, and even neurobiology.
Excellent. Well, with that, I'd like to thank you, Sheela. It's been very informative. We learned quite a bit about microbiome research, automation, and emerging technologies, and the QPix FLEX and what it can do for researchers in this field. So I want to thank you. I want to thank our audience for attending and let them know that the full webinar video will be available at the webinars dropdown on the LabX homepage. Thanks very much, Sheela, and you're welcome back anytime.
Thank you so much, Damon.
Take care.